Welcome back, everybody. This is a video lecture on protein regulation. The word regulation can be substituted with the word control. So how proteins are controlled. So let's get started. The agenda is a quick recap of the roles of proteins, and then we'll look at how proteins are controlled at various levels in enzymes, metabolic pathways, phosphorylation control, Gprotein regulation, motor proteins, and protein machines. Then we'll have a quick synopsis of protein research using electrophoresis and mass spectrometry. And then we'll finish off with a little discussion on protein families, how proteins have evolved, especially with respect to domains. So let's go on to the next screen. And here we have a quick review. So proteins are workhorses of the cell. It doesn't mean that they do everything for the cell, but they do most of the work at the direction, in most cases, of the nucleic acids. So DNA and RNA, they regulate how proteins are going to do their jobs, and they're also responsible for making the type of proteins that are necessary within that cell. So almost all functions are performed by proteins. Information storage is mostly the domain of nucleic acids, but almost everything else is left to the proteins. Although, as you'll see in later lectures, RNA still is involved in some very critical steps that are still not handed over to proteins. So we're going to talk about RNA and its role in many critical steps of cellular regulation. Proteins can be broken down into two functional structural groups. So the structural proteins are anatomical in nature. They provide a scaffold, such as the cell cytoskeleton, or the support for the membrane, or the structure of the nucleus, support for the chromosomes, etc., etc. And there's another class of proteins known as functional proteins. And these are physiological proteins that perform chemical reactions. They partake, as you'll see today, in many regulatory mechanisms within the cell. So from physiology and anatomy, we can apply these two terms to the roles of proteins. So proteins are very important. Let's turn our attention and look at the regulation control of enzymes. As we know from the previous video lecture, you can have a substance A inside the cell, and you want to convert that to substance B. In almost every case inside the cell, an enzyme is going to be responsible for catalyzing, i.e. speeding up that particular chemical reaction. So the enzyme would sit here somewhere, like that. So every step in a biochemical pathway has an enzyme responsible for catalyzing that step. A chemical pathway can be quite complex, but in the example that we're going to study right now, let's keep it pretty simple. And we're going to look at how enzymes are involved in regulating the one step that they're concerned with, and in some cases, additional steps in the metabolic system. So we're going to focus on this slide, on this one enzyme down here. So let's get rid of some of the other stuff that we don't need for now. We'll bring it back when it's necessary, right? So let's take this enzyme, make it nice and large, so it's obvious what we're talking about. So here's the enzyme. Oops, let's draw that again with a different pen. Here's the enzyme. And suppose there's a site on the surface of the enzyme at which the chemical reaction takes place. And we will call that site by its proper name. It's called the active site. There you go. So the substrate comes and lands and fits into the pocket at the active site. So what's important for that to happen, and how does the enzyme control that behavior? Well, the enzyme is only going to fit one or a few related shapes of molecules. So the enzyme is going to be very specific to its chemical reactions. So if the object comes and sits down in here like this, the substrate of the enzyme, then there's enough time for the enzyme to perform some contortions and change the substrate into a product. For that to happen, the enzyme has to have a particular shape, and if that shape is altered by some external factors like pH, temperature, salt concentration, or even a mutation at the gene that produces this enzyme, that's going to affect the shape, and it's going to affect the control over that pathway from step A to step B. Okay, and the other thing is charge. The charge on the surface and within the pocket of the enzyme has to be somehow compatible with its function. So normally what we find is, in order to guarantee a tight fit between the substrate and the active site, the active sites will have corresponding charges to the charges on the surface of the substrate. In many cases, not all cases. So that guarantees an even better fit between the substrate and the enzyme. The product will then leave the active site, and then another substrate will come in. And some enzymes can perform 10 million reactions per second. So 10 million times the substrate can swap in and out. So that's amazing. Let's get back to the control over this enzyme. So what can control this enzyme? Well, the temperature of the cell. So as the temperature increases, the movement of the chemistry takes place faster and faster, and the reaction speeds up. But up to a certain point, when the temperature becomes too high, the enzyme will change shape. And if it changes shape, it may become non-functional. Other things that can change the functionality of the enzyme would be a pH. If the environment becomes too acidic or too alkaline, that too can cause a reshuffling of the charge within the enzyme, causing it to adopt a new shape. And likewise, salt concentrations. And there's many other factors that can control the enzyme. Those are all physical entities. Let's look at something to do with the enzyme itself. Most enzymes have one or more sites other than the active site at which the activity of this enzyme can be regulated. So let's put that in place. We need our eraser. Let's erase a small part of this and change that into something different. And we'll keep the same color. So let's put that as this. So now we have a new site down here at the bottom left-hand side of the enzyme. And this can fit another ligand, right? A ligand, remember, is anything that binds to an enzyme. So the substrate up here will be a ligand. And this other molecule, let's give it a shape. Let's make it into a nice rectangle. This molecule here, let's call it L for ligand. This molecule here can come and fit into this cavity on the side of the enzyme. This space here is called the allosteric site. Now the word allo means other, steric means shape. So it's not the active site, but it's another site. So allosteric site, and that's the word down here. So an enzyme can have one or more active allosteric sites on its surface. The binding of this ligand to its binding site can have a knock-on effect within the shape of the enzyme that causes the active site to change shape by rearranging its molecular interactions. If the active site changes shape, then it will give it a new configuration that in most cases will make it less efficient at binding to its substrate. Therefore, by binding the ligand down here, you're going to change the activity of the enzyme up there. And if it slows down or eliminates its active site, the binding site for the substrate, then the enzyme is being regulated by the ligand down here. So this indirect association between molecules is very common, more common than you think. Almost every enzyme that we know has an allosteric site, at least one, to which it can bind various chemicals from its environment. So this is a very powerful mechanism for controlling the behavior of our said enzyme. Okay? So let's put that in a larger perspective. Suppose we wanted to slow down the conversion of A to B by this enzyme. And in this case, A will be the substrate that binds to the active site. Well, we can modulate that. The word modulate simply means control or regulate the behavior. We can make it go faster, or we can make it go slower. And we can also change it by certain amounts. So we can make it go much faster or much slower. We can even stop it from happening. And all that can be controlled by the ligand binding to its allosteric site. So this is a very, very precise way of controlling within small increments, percentages, the behavior of this enzyme. Most people think of enzymes as either being on or off, like a light switch. But the better analogy is to think of an enzyme as a dimmer switch that can be controlled between off, which is 0%, up all the way to 100%. And in some cases, you can push it beyond 100%. You can have a super bright light where the dimmer switch is put into overdrive, say 150 to 200%. So it looks like every enzyme that we know of has the potential to be regulated in that fashion. Okay, so this is very important. As you'll see, enzymes can set up local circuits. That means they don't need to report to the nucleus. They don't need to report to any major organelle within the cell. Everything is done at the local level. So within this vicinity of the cytosol or the nucleus, decisions will be made automatically by the concentration, in this case, of the ligand. If the ligand concentration increases, it's going to be more likely to bind to the allosteric site, therefore regulating in a different way the active site. And as the concentration of the ligand falls, then it will fall off more likely than not, and that's going to restore the active site to its wild type configuration. So this is a local circuit, and most enzymes do work at a local level. That saves the cell from having to make these minor decisions on a second by second or even millisecond by millisecond basis, right? So we say that enzyme regulation is self-regulated with its chemistry. You'll see a better example on the next slide. But the enzyme is sensitive to physical and chemical conditions at a universal level, such as temperature, pH, and salt concentration. So keep all that in mind, please. So let's look at a wider picture of the same system. So we're going to write down our metabolic pathway again. So we have A going to B, and remember, these chemical reactions can go in either way. So we have to put a double arrow there. So B can be converted to C, and C can be converted to D, and we'll stop at E, although this can become quite complex. So E is the final product of this pathway, which starts with A. A could be some kind of food molecule, for instance, that enters the cell and is then converted to something step by step until it reaches E, all right? And as we said earlier, every single step in this pathway will have its enzyme. So E1 will be responsible for the conversion of A to B and B back to A. And then enzyme 2 will be at that step, enzyme 3, and then finally enzyme 4. And again, each one of those enzymes has an active site to which it binds the substrate, and then it releases the product. Let's introduce our E1 shape. The enzyme E1 had a particular shape on the previous slide, if you recall. It had a shape very similar to that. So that was on, this is the enzyme that we're talking about now, okay? So there it is, drawn in miniature. So A would bind here, like that, and then be converted to B in this instance. What about the allosteric site down there? What will that bind? Well, in this case, it's going to bind E. Okay? So as the concentration of E builds up inside the cell, either in this location or another location, E will diffuse through physical attributes into the region where enzyme A is operating. Now, there may not be just one copy of enzyme A, there may be a number, maybe hundreds, thousands, but they're sitting there converting A to B. As the concentration of E builds up, it will then resonate, vibrate, in and out of the active site. Wrong word. It will resonate in and out of the allosteric site. So E will come and bind in here, like that. So that's E. And E will sit there and change the behavior of enzyme E1. And E1 will then respond in some fashion. And the most common mechanism is listed right here. So it's called negative feedback, or negative control, or feedback inhibition. Take your pick. It all means exactly the same thing. So this little circuit can control itself. If it produces too much E, then the concentration of E increases inside the cell, and then that drifts across, and it starts binding to the allosteric site on enzyme E1. Therefore, in this case, because it's negative feedback, it will start shutting down the activity of enzyme E1. So the analogy that we give you is this is a conveyor belt producing some kind of food, right? So the ingredients go in at the beginning, and then they get mixed by the conveyor belt. And at the end of the conveyor belt, you have cupcakes, right? Let's pretend they're cupcakes. And if you're not selling the cupcakes, they build up inside your warehouse. And as they build up, they start taking up more and more room. And eventually, there'll be so many cupcakes inside your factory or your warehouse that they'll come back and interfere with the conveyor belt. Therefore, shutting the conveyor belt down until you start selling your cupcakes. So this is a very logical circuit. So this is what we mean by local circuits, right? So local circuits are more common than you can imagine, and they're everywhere. In fact, most systems are local circuits. And they're self-regulating, and they are sensitive to other things, temperature, pH, salt concentration, and sometimes the concentration of other chemicals too. So this is a negative feedback mechanism where E has a negative effect on enzyme E1, therefore slowing down the pathway. This is the most common type of biochemical pathway that can operate inside a cell. Of course, this is a very simplified circuit. There are more complicated circuits than this, but we have to start somewhere in understanding these. So hopefully that makes sense to you. So this is a way of regulating the behavior of enzymes, right, in these pathways. Okay, so let's move on and talk about phosphorylation. So phosphorylation involves a particular atom, let's put it like this, and give it an oxygen here and an oxygen there, right? So that's called a phosphate, or a phosphate will be then missing this last oxygen. So let's take that out, because then it can be used there with that bond to bond to something else. So you can see by the charge, it has a very, very negative charge along the molecule. And that particular structure is able to then impart on other things some of that negativity. And that's what we do. We call this a chemical tag, right? By adding this chemical tag to another molecule, we can change the shape of that molecule and the charge distribution of that molecule. In that way, we can either make that molecule more active, or we can make it have a different shape. So we're going to look at both scenarios. So in the top green one, we're going to look at the shape, and in the bottom one, we're going to look at the activity, right? So suppose we have something inside the cell. Let's call it A. So this substance A has a particular shape, and suppose that it's active at this point, right? It's doing its job, or whatever it is inside the cell. And we want to change that behavior. We can take another enzyme called a kinase. What kinases do, they add phosphates onto other molecules like that. So now that phosphate belongs to the surface of A, and it will then have a different shape, and that charge will then affect the shape of A, and that could change the configuration of A, therefore turning A from an on configuration now to an off configuration. So you've turned off A in its activity, whatever it was. So if A was an enzyme, you basically turned it off, right? If you want to turn A back on again, all you have to do is remove the phosphate group from the end. Now, one word of caution, right? Normally the kinase can't do both. The kinase can add on a phosphate, but it can't remove it. Nature has thus invented another enzyme called a phosphatase, and the phosphatase is able to break that bond and remove the phosphate, therefore restoring A back to its original configuration, right? So that's what a phosphatase would look. So phosphatases remove phosphates from their target molecules, and kinases add phosphates to their target molecules. And you may not think this is that important, but let me tell you, there's over 800 different types of phosphatases and kinases that have been identified inside cells. So we have 22,000 protein coding genes, and almost 1,000 of them are involved in this particular regulatory system. So the inside of a cell is packed full of various types of kinases and phosphatases, right? And their activity is controlled by local circuits, as we learned on the previous slide. Okay, so this is a great way to regulate in quick time. You can do this many times per millisecond. So you can change the behavior of A quickly and rapidly. I think those words are the same, are they not? So that's one way you can control the activity inside a cell, right? The other way is through phosphorylation control. It's very similar to what we said in the above example, except now we're going to talk more about shape. So suppose we have a substance B, and B looks like a rod. There you go. And B can be phosphorylated, say 3 times, right, by a kinase or some other enzyme. So we can change the shape of B from a smooth cylinder like that to something that contains, say, 3 phosphate groups. So here will be a phosphate, another phosphate, these molecules here. 3 of them would be added. And now we see that the cylinder has 3 bumps at particular points along its length that it didn't have before. What are the consequences of that? Well, maybe there's another protein inside the cell that's waiting around to do a particular binding to the cylinder, but it can only bind to the cylinder if the cylinder has the 3 phosphates on its surface. So when the phosphates were not there, this protein over here, let's call it X could not bind to the cylinder. But now the cylinder has been phosphorylated 3 times, and it has that new shape. And that shape is compatible with X. And what X will do now is come and bind to the surface of the cylinder. And that binding may change the shape of X. And now Y, which is sitting around the cell looking for a target, now Y can come and bind to X, whereas it couldn't bind to that because it didn't have the cutoff on the edge. So this cascade of binding is quite common inside a cell. So if something triggers a shape change, normally phosphorylation, then the binding of other substances and their activation takes place. And then that binds and activates something else until the cell behavior is changed. So phosphorylation control is very important, almost as important as kinases and phosphatases. So remember that too. Let's go on a bit further, a bit deeper. Let's talk about Gprotein regulation. So this is a more sophisticated version of what we just saw on the previous slide. So just a warning, cells have evolved 2 major classes of G proteins. We have the small G proteins, and they're the ones we're going to talk about next. And then we have the large G proteins, which normally come in 3 subunits. We don't talk about them at this time. They are discussed much later on in the video lectures, right? And there may be a copy of one of those lectures already on YouTube. So if you can't wait, you can watch that if you wish. But it's not covered in this video lecture. So we're going to stick to understanding how small G proteins work. So how do we explain this? Well, if you think of a partnership, right? So it's like the algae living inside the animal that makes coral. So that's a partnership. So let's draw the protein first in one color, and then we'll put the partnership in there, and we'll explain what the partnership is. So let's use red to represent the protein. So here's our protein. Then it has an active site down there, and then it could have a couple of allosteric sites. So that's our protein. Now the protein has a partner. Let's put the partner in green. And our partner will be like this. So it'll be a nucleotide, because that's what the G stands for, right? We know that there are 4 types of nucleotide in DNA and RNA. And so adenine, guanine, cytosine, and thymine. So the G stands for the guanine, right? So the guanine nucleotide is composed of a sugar, a base, and phosphates. So normally there are 3 phosphates like that. And that's called a GTP. Just like ATP, right? But this is made of a different base. So the GTP can then be converted to GDP when it loses a phosphate. And that will change its shape and its charge. So sitting in the pocket right now, the partner is 1 molecule of GTP. And when GTP is sitting in the pocket, that will activate the enzyme. So the active site here is now going to be functional, and it's going to be converting A to B at some rate. So the G protein, the whole thing is called a G protein now, the green and the red. The G protein is now active. So the G protein is on. Okay. Here's the kicker. Nature has given the green GTP an option of cutting itself. Yes, it can cut itself. At a predetermined time in the future, maybe a millisecond or maybe a second or so, the GTP will become GDP, and it will lose one of the phosphates. So let's use a different color for that. So at some time in the future, not long from now, this GTP will become GDP, and that phosphate will be lost. The loss of the phosphate you may not think is much, but it's going to change the configuration of the active site so that it no longer can bind to its substrate. So now the G protein has been turned off. It's off. The GDP, which is now the double phosphate here, that will then fall out because it can no longer sustain itself in that cavity. And once it falls out, you get to this situation. So let's use a small eraser to get rid of this, so we can show you what happens, right? So now the system is actually off, right? Because it goes off when you go from GTP to GDP. And the system stays off as it ejects the GDP. Now that site there is able to bind a fresh molecule of GTP. And the GTP will come in and it will sit down as it was previously with its partner. And that will turn the system back on again. So you can see we have a self-timer. A protein that times itself to be on and off, on and off, on and off. So that's what G proteins can do. There's one extra step that I didn't tell you about, right? So the extra step is the binding of GTP. If that has to be triggered by something else, then let it be. That will introduce an extra step into our circuit, right? So let's get rid of one of these steps. Let's get rid of this. And now we can add that in there too. And that makes it into a more sensitive system. So an event has to take place before a fresh molecule of GTP can bind back into this site. Therefore, the protein will be off until that event happens. So that could be a sparkle of light hitting your eye, you know, something like that, or somebody touching your skin, right? When the event happens, the GTP binds back to its G protein, and then the system is turned on, and that is then communicated for a brief period of time. Then the system resets itself by ejecting the GDP until the next event happens. So this is a fantastic adaptation of proteins to conditions. It's amazing, okay? So this partnership is a molecular timer that keeps the G protein on for a certain duration, and then it resets it to the off position until the next event. Just a word of caution, because we've found so many of these, over 200, we're finding that they fall into two types. Most of them are turned off when they lose a phosphate, but some of them happen to be, unfortunately, the other way around. They're turned on when they lose a phosphate. So just be careful if you ever come across an example like that in the future. It's amazing, right? This is one of my favorite things to talk about. So let's move on to another favorite of mine, motor proteins. So motor proteins are proteins that can generate a force, right? They can generate a force. And the way they normally do that is slide over another protein. So you're familiar with this maybe from high school, and it's the way that our muscles work, okay? So in the muscles, we have two types of protein. We have this type of protein, and then we have, let me put a different color. Then we have another type of protein that has hooks on it like this, okay? And there's another one on the other side like that. So when a nerve impulse is triggered from your brain, it travels to your muscle and it tells the muscle to contract. Through a very sophisticated chemical integration, chemistry is released, and that chemistry uses calcium, and the calcium causes the two types of proteins to interact. And what happens is that the red ones, they start walking along the green one in both directions. So this red one comes in, that red one comes in. And they come in from both sides, and that shortens your muscle, therefore you can lift something up. So these are called motor proteins. They use a lot of ATP to cycle through this type of movement that they make along the other protein like that, okay? So this is one example. My other example is one that you see a lot on the internet. You have from the nucleus emanating a cytoskeleton. So these are protein cables that are emanating in various directions within a eukaryotic cell. And what we do find are there's some walking proteins that actually walk like that, and they carry a cargo with them. So this could be something full of sugar, like a starch vacuole, or a neurotoxin, or a neurotransmitter, whatever. And this protein here, the one in red, the one with the legs, is able to take steps like that and walk along this cable, taking that cargo to its destination. So those are called walking proteins, right? And if you don't believe me, just look on the internet under cell animations, walking proteins, and you'll find a video by Harvard Medical School. That's beautiful to watch. Okay, and of course, just like the cousin up here, they do require the hydrolysis, the breakdown of ATP to release the energy for at least one of the steps that it needs to take, right? So it takes steps based on the availability of energy in the form of ATP. So these are called motor proteins because they can cause mechanical changes, either at the local level, i.e. walking, or at the cellular level or the tissue level, i.e. muscle contraction. So these are fantastic as well. Oh yeah, I have another favorite now, protein machines. So what we're saying now in this particular instance is that nature has evolved so much that it's put components together. So you imagine your car, right? So your car is a machine. It has an engine that has to work. Then it has a radiator that has to keep the coolant cool. Then you have the electrical system that has to power various things. Then you have the wheels and the steering wheel, the seats, the mirrors, the windows. Everything has to work together. So your car is a machine. Just like a machine that your car is, we can have machines that are made of protein parts inside cells. So the classic one that comes to mind is your ribosome. Your ribosome, and we can talk about ribosomes in the future, your ribosome is a machine. What does this machine do? It's a sewing machine for making polypeptides. That's what ribosomes do, right? So one part of the ribosome reads the messenger RNA, right? So the messenger RNA will be down here. Another part of the ribosome brings in the amino acids by interacting with something called tRNA or transfer RNA. And another part of the ribosome puts them together. And another part of the ribosome knows where to start and where to stop. So the ribosome is a machine. As many proteins working together, doing the right thing at the right time. So the example of a sewing machine is pretty good. I like that, right? But there's many more machines inside your cell. So the product of one protein becomes the substrate of a protein that's tethered to the back of the first protein, right? So that makes sense. If you have 3 proteins inside the cell, like this, A, B, and C, and what A does, it takes X and converts it to Y, and then B picks up Y, converts it to Z, and Z converts that to, let's say, P, right? If these proteins are working in harmony together and they don't do much else but except help each other, then it makes sense to put them all together into a blob so that A, B, and C are working as a machine. So when A finishes something, it's right there to be picked up by B to save time. And then when B finishes and produces Z, Z is ready right there to be picked up by C. And then the product is released very quickly. Instead of me having to drift around the cell to find B, B is tied or tethered to the back or the side of A. So the whole thing would be called a machine because it's a multi-part protein machine. Amazing, right? There are many machines inside the cell, and you'll see a lot of them in this course. So the next one that's coming up would be your DNA replication machine. Yes, it's the machine that copies your DNA during the S phase of the cell cycle. Amazing. All right, let's move on then and look at protein research. How do you investigate which proteins there are in a sample? How do you isolate the proteins? How do you characterize the proteins? And how do you figure out what they're made of? Just a word of warning now. Some of these techniques are aging. They're getting old. We have powerful computers that can be taught how to model proteins simply from giving them the DNA sequence, right? Remember, proteins are made by ribosomes, but the ribosomes are just machines. They need their instructions. And their instructions ultimately come from the DNA of the cell or the mitochondria or the chloroplast, right? Get that in there. So made to order proteins. So if you know the sequence of the DNA, you can then, using current day knowledge, work out what the protein will look like without having to do these sophisticated procedures. But sometimes you do, because that's the only thing you have, right? We don't have enough knowledge about the DNA yet. So electrophoresis is basically a mechanism of separating protein based on certain characteristics. The most common one is going to be the size, right? So you could have a protein that has a polypeptide that long. You could have another protein that has a polypeptide that long, and you can have another protein that has a polypeptide that long. And they're all within the same sample taken from a cell or something. And you want to know how to isolate the small one away from the other two. So what you do, you use some reagents to neutralize the charge along the proteins. And the reason for that is different proteins can have different charge along their length that may not be present on all proteins at the same level. So you use various chemicals to neutralize that charge, right? So they coat the surface and they neutralize the charge. So now each segment, each amino acid, each monomer of the protein is equivalent. So when you do run them on a gel, what is a gel? A gel is normally made of either one of two substances. It's either made of extracts from seaweed called agarose, it's a carbohydrate, or it's made of a more dangerous polymer called polyacrylamide. It doesn't matter, right? So the gel is simply a slab, and inside the slab we have little cavities called wells. And what you do, you take your sample, once it's processed, you drop it into the well, and then you apply an electric current, right? And the electric current will then pull the protein through the gel at a proportional speed. Now, I told you that before that you neutralize the charge on the surface of the proteins. Yes, but then you can also introduce a standard positive charge all the way along the top of that, so that every part of the protein now carries a standard charge. So that will now be pulled towards the negative if it's a positive charge on the surface of the protein, and then the fragments will be separated according to size. So the smallest fragment will travel the fastest through the forest of sugar trees. The medium-sized fragment will travel intermediately, and the large fragment will be hindered by the forest, and it'll slow down and get trapped, and it'll be delayed. So now you've separated your sample according to, in this case, size, okay? And there's many ways of separating proteins, but this is just one of them. And of course, you can do the same thing with DNA, right? So DNA is negatively charged without any further treatment, it is. And in that case, you would have to reverse the poles. This would have to be positive, and this will be negative, and the DNA will travel again according to its size along the gel. So these gels are very common. So when you do use DNA, that's called a southern blot, yes, or southern electrophoresis. When you use a protein, that's called a western blot or western electrophoresis. Okay, just a minor thing. So what about mass spectrometry? This is a mechanism for using X-rays on crystalline protein structures. So you get some protein from some source. If you can convert it to a crystal, great. Now you can put it in a machine where you bombard it with X-rays, and the pattern of X-rays that are picked up by the detectors and then transmitted to a computer will give you an image. And from that image, either the software or the scientist can determine the placement of atoms within the crystalline structure of the protein. And some very clever people can then work out where each amino acid is within that structure and which amino acids are where. It's amazing, okay? So again, like I said, these days we can use DNA structure in most cases to predict what shape the protein will have. But sometimes you can't do that, either because you don't have the DNA sequence yet, or it's impossible to predict under certain circumstances what the protein will look like. So that is called, the structure of a protein can be determined through that mechanism. Let's just finish off then by talking about domains, right? From a previous video lecture, we told you that the primary sequence of a polypeptide is a string of amino acids. And then that's the primary sequence. Then the secondary sequence is where it starts folding up either as beta sheets or alpha helices, right? And this will be a secondary structure. That's a secondary structure. And then maybe another secondary structure here. And this is this molecule here beginning to fold. Then the tertiary structure is where this chain begins to collapse on itself like this to form the final protein, okay? And in some cases, and the last step is optional for some proteins, in some cases, you'll get this combining with another one to form the finished protein. But it doesn't matter, right? The word domain simply means a part of that polypeptide that is responsible for doing something. In most cases, this thing here carries one domain, right? The whole thing, the whole subunit of the whole protein is only able to do one thing, i.e. bind to DNA. So one type of domain is a DNA binding domain, right? That's all it can do. It binds to the DNA and does some activity. Other types of proteins have domains that are not DNA binding. They could be something else. So we have other things that can bind to other proteins, right? So protein binding to protein. So that domain will be a protein binding domain. Other domains can act as enzymatic or enzymes, right? So there could be an enzyme with an active site and allosteric site. So proteins can be classified according to what they do. And what they do is based on domains. So the domains, if you study enough proteins, they fall into these buckets, right? But sometimes, and in more recent evolution, i.e. us and animals and plants and multicellular creatures, so more evolved creatures tend to take proteins with single domains and combine them to make a dual functional protein. So a protein with two domains, right? So this happens through gene mutation, where one gene and another gene somewhere else with a different function, they are brought together through mutation, and now they make a giant gene. And the beginning of the gene has the information for one domain, and the end of the gene has the information for a different domain, right? And now you have a protein that's produced that has two domains. Maybe this one binds to DNA, and that one there has a enzymatic function. So one side of the protein will bind to DNA, and the other side will perform a chemical reaction. And from there, you can go even further. You can have proteins that now have 3 domains, or 4 domains, or 5 domains, right? So once single domain proteins were evolved, rather than making new single domain proteins from scratch, Mother Nature doesn't like to do that. If she already has a recipe, she's going to modify that recipe to make something more wonderful. And that's what she's doing here, right? She's taking existing proteins and then mixing them together to make even more functional multi-domain proteins. So new protein domains seem to be more commonly evolved from existing infrastructure that Mother Nature has. So everything we've done today has been looking at various ways in which proteins can be regulated, i.e. controlled. So thank you so much.